Hello everyone. Today we are going to discuss about gene pool. Let us first get started with the definition. A gene pool is the total collection of all genes and their alleles present in all individuals of a sexually reproducing population at a given time. It represents the genetic makeup of a population, not of an individual. Larger gene pools is equals to more genetic diversity. Smaller gene pools is equals to less diversity. Higher risk of genetic orders. Some of the examples are: consider a population of 100 beetles. Gene for body color has two alleles. Dominant one capital B black and small b brown. If all beetles together contain 120 alleles of capital B and 80 alleles of small b, then the gene pool consists of capital B and small b alleles with their total numbers. Now let us talk about gene frequency. Gene frequency, also known as allele frequency, is the proportion of a particular allele in the gene pool of a population. It is expressed as a fraction or percentage. The formula for this is gene frequency of an allele is equals to number of that allele divided by total number of alleles of that gene. Example: in a diploid population of 100 individuals, total alleles for one gene is equals to 200. Allele capital A is equals to 120. Allele small a is equals to 80. Frequency of capital A is equals to 120 by 200, that is equals to 0.6, and frequency of small a is equals to 80 upon 200, that is equals to 0.4. So here P plus Q is equals to 1 and 0.6 plus 0.4 is equals to 1. Now let us talk about Hardy-Weinberg principle. The Hardy-Weinberg principle states that allele frequencies and genotype frequencies in a population remain constant from generation to generation in the absence of evolutionary forces. This constant state is called Hardy-Weinberg equilibrium. Hardy-Weinberg equation. For a gene with two alleles capital A and small a, P plus Q is equals to 1. Genotype frequencies P square plus 2PQ plus Q square is equals to 1, where P square is equals to frequency of 2 capital A, 2PQ is equals to frequency of capital A and small a, Q square is equals to frequency of small a. Example: using P is equals to 0.6 and Q is equals to 0.4. P square is equals to 0.6 square, that is equals to 0.36. 2PQ is equals to 2 into 0.6 into 0.4, that is equals to 0.48, and Q square is equals to 0.4 square, that is equals to 0.16. Total is equals to 0.36 plus 0.48 plus 0.16, that is equals to 1. This population is in Hardy-Weinberg equilibrium. Now the question arises that how Hardy-Weinberg equilibrium is maintained. Hardy-Weinberg equilibrium is maintained only when the following conditions are fulfilled. First, large population size prevents genetic drift. Small populations show fluctuations due to chance. Second, random mating. Individuals mate without preference for genotype or phenotype ensures equal chance of allele combination. Third, no mutation. Mutations create new alleles altering gene frequencies. For equilibrium, mutation rate must be negligible. Fourth, no migration, no movement of individuals into or out of the population. Migration introduces or removes alleles. Fifth, no natural selection. All genotypes have equal survival and reproductive success. Selection favors certain alleles changing frequencies. Sixth, significance of Hardy-Weinberg equilibrium acts as a null model for evolution. Any deviation from equilibrium indicates evolution is occurring. Helps study natural selection, genetic drift, migration, and mutation. In summary, gene pool is total alleles in a population. Gene frequency is proportion of a specific allele. Hardy-Weinberg equilibrium is constant allele frequencies maintained by large population, random mating, no mutation, no migration, and no natural selection.